Okay, in the last video we looked at this concept of the inner terrain, looked a little bit at the history and how what used to be referred to as the vis medicatrix in the humors was essentially resorbed into the concept of milieu intérieur and homeostasis, and how it's the neuroendocrine immune system, which are the three primary systems which maintain homeostasis. Let's look specifically now at the endocrine system, glands, and hormones. Look at a concept known as hormesis, which is going to be very important for understanding endocrine action. Also look at classes of the hormones and their receptors. I'm going to introduce a concept known as the neuroendocrine centers, and then look at the regulation of hormones in the blood, and then again end on the hypothalamus. So let's talk about glands first. So there are two major types of glands. There are extrican glands, which again have ducts that carry their secretory products, usually their peptides, to a surface, like a mucosal surface. And this would be the extrican pancreas, for example, would be a good example there. An endocrine gland, on the other hand, would be a ductless gland, and they secrete hormones directly into the blood. So scattered throughout the pancreas are the islets of Langerhans, and the beta cells within them secrete, for example, insulin, which goes right into the blood. Now, the same concept applies to organs as well. So if you look at the liver, we can think of the gallbladder system, the biliary system, as the extrican part of the liver, versus the liver itself actually is able to put substances into the blood directly. And so the line between what we call the classical endocrine glands, like the thyroid and the pancreas and the pineal gland, is getting blurred a bit because we're realizing a lot of organs also secrete, like endocrine glands, directly hormones into the blood. So we'll have to talk about those as well. But the classic glands include the pineal gland up in the brain, the hypothalamus and pituitary gland, spent a lot of time on that, thyroid and parathyroid glands, the pancreas, including the endocrine pancreas, the adrenals, and then the gonads, and that would be testes in males, ovaries in females. So that's the classic endocrine glands. But again, we can think of secondary endocrine glands. Non-classic glands would be the intestines. So the small intestine secretes a whole range of peptides, GIP and GLP-1, cholecystokinin, secretin, amylin, etc. And all of these have major roles on regulating systemic metabolism, and that's really an active area of research right now. The liver also puts out hormones. They're called hepatokines, and interestingly, when the liver becomes too filled with triglycerides, fatty liver, it begins to put out a lot of these hepatokines, which actually can induce insulin resistance in peripheral tissues. And so that's where a stressed organ can actually influence the whole body here. Now, if we back up again and say, why did all that triglyceride accumulate in the liver? Well, interestingly, high insulin often drives that process. So here we have the endocrine system regulating the organ system. Kidneys secrete hormones, so vitamin D, for example, calcitriol, the 125 dihydroxy vitamin D, this is a hormone. This is not a vitamin, it's a hormone. The D3 you take at the store, from the store, you have to essentially convert that to 125 hydroxy vitamin D in the liver, and then the kidneys hydroxylate it one more time, and that's the active vitamin D. So we can think of vitamin D as an active kidney hormone. Erythropoietin would be another, and that stimulates your red cell production and hemoglobin production in your bone marrow. And then the renin, of course, is another kidney hormone. The heart secretes hormones, atrial natriuretic peptide, ANP, as well as BNP, and these regulate fluid balance. They tell the kidneys to initiate diuresis if there's too much fluid volume in the blood. Immune cells also release hormones, but more specifically cytokines. The thymus gland, we usually think of the thymus as atrophying, after you're a teenager. But we now know that the thymus continues to secrete peptides called thymopoietins. And these talk to your lymph nodes, and they actually help T cells prime better, mature better, when they are faced with infection. And so the thymus, interestingly, is something that continues to play a role, and that is very important for proper immune health. Interestingly, we know that the thymus actually undergoes a second senescence, really, after around age 65, and that's the point when people are now much more susceptible to cancers, but also infections. And so could that be because of a dwindling thymic activity? Still, that's something that we don't fully know yet. Placenta secretes hormones, and then adipose tissue secretes what are called adipokines. And adipokines are essentially cytokines from adipose, which can regulate systemic metabolism. They could feed back to the liver or the brain to influence appetite and so forth. So that's a little bit about the different glands. Now, embryonically, the glands really come from all three germ layers. So from the ectoderm, remember that forms the nervous sensory system. The adrenal medulla and the pituitary come from the ectoderm. The mesoderm, really this forms the adrenal cortex and the gonads. And the endoderm, which is really the source of your metabolic digestive organs and activity and mucosa, this forms the pancreas, the thyroid, parathyroid, and thymus. So the germ layers really go into forming all the different glands. Now, hormones, these are chemical messengers that enter the blood. They're carried in the blood to target cells, and many hormones actually have to be bound to plasma proteins, as we'll talk about here shortly, to be able to be carried in the blood. They act on specific cell receptors that are either on the surface of the cell, or in the case of steroids, they're actually in the cytoplasm or even the nucleus. They are, in general, hormones, their receptors are more slowly and longer acting than neurotransmitters. So think of neurotransmitters as very fast integration of communication, and the hormones are a lot slower. Really, the actions are diverse, and we talked about these already a bit. But hormones are the prime regulators of metabolism. They stimulate or inhibit growth, regulate your reproductive cycles, regulate other hormones, mood, etc. Now, we can also talk about autocrine agents. These are chemicals secreted by a cell that acts on the same cell. So there's a receptor on the cell for it. Versus paracrine agents, that's a cell, and if here's some nearby cells, this cell can talk to nearby cells. Again, a hormone's different in that it goes throughout the entire body, communicates with all cells. Now, there's going to be a difference, of course, in the brain with the blood-brain barrier. Not all hormones can get across that barrier, and so that can be an issue. But usually lipid-soluble hormones, like the steroid hormones, can get across that barrier. And we now know that the brain is highly dependent on your adrenal and sex hormones for proper development and function. So we'll be looking at that in a lot of detail when we look at the adrenal and the sex hormone section. Now, the concentrations of hormones in the plasma, I think, are really interesting. We usually talk about, a lot of hormones are in the range of picograms, which is 10 to the minus 12, which in the homeopathic language is a 12x dilution. And then 1 nanogram, which is 10 to the minus 9th, and that's a 9x dilution. Now again, a homeopath would say you have to dilute a substance, but also succuss or shake it at each point to make a remedy. Well, interestingly, as hormones are released, they're released often in a pulsatile fashion. They're succussed in the blood. Very interesting. So it's almost like the body potentizes these substances to be active. And these incredibly tiny dilutions have profound effects on body function, body form, your morphology, and so forth. So melatonin general ranges are between 10 and 100 picograms. Growth hormone in adults is usually under 10 nanograms per milliliter. ACTH, which is the hormone from the pituitary to stimulate your adrenal glands to secrete cortisol, is again in the picogram range. Thyroid hormone, free thyroxine, is in the nanogram per deciliter range. Insulin, picomoles per liter. Estradiol, picograms again, and then testosterone, nanograms. So very tiny amounts of hormones. And that's why when we see people taking large amounts of hormones, you have to be careful when you replace hormones that we try to achieve physiologic levels, not over that. That's going to be a very important concept we'll talk about here. All right, so I just want to point out how dilute many of these substances actually are. Now, a very important concept we're going to need to understand, not just for endocrinology, but for medicine in general and therapies, is something known as hormesis. And this refers to what we call biphasic dose responses of different things like hormones or a drug, an herb, etc. You might have heard in the literature something known as a J-shaped or U-shaped curve. This has an interesting history. So Hugo Schultz, was a German pharmacologist back in the late 1800s, and Rudolf Arndt was a German psychiatrist who worked with Schultz, and they came up with what's called the Arndt-Schultz rule. And this really described this phenomenon that very low doses of a substance, and they looked at, for example, phenol, which is toxic, and its effects on yeast cultures. That was one of the first experiments. They found that very small doses of phenol actually would stimulate yeast growth. And then if you increase the dose of phenol, we started to see the opposite. So as you increase your dose response, you begin to see an inhibitory effect on physiology versus small doses have stimulatory. We can use the same curve to actually understand things like vitamin C. So at 0 vitamin C here, we're going to have a problem. You can't make vitamin C, so you're going to have a deficiency. Now we start to increase the vitamin C in the system, and we see that there's a stimulatory effect on physiology. But lo and behold, you take more C, what happens? Boom, you start to suppress. So in cancer therapies where we use a lot of vitamin C, we actually are using it for a pro-oxidative effect, more of a inhibitory effect on cells, not a stimulatory effect. Now, this will be very important because we'll actually see in medicine there are two different approaches. One approach is to use substances to stimulate physiology. The other is to use it to suppress physiology. And this is, I think, an area of a lot of confusion. So if we look at a lot of heavy metals, it was discovered by later researchers. So Southam and Ehrlich, they looked at biphasic dose responses in plants. But a modern researcher is Edward Calabrese, and he's done a lot to really put hormesis back on the map. It was kind of dismissed by conventional pharmacologists who usually, when they start researching drugs, they start at these dosage ranges that are already out here. And so they were missing these very small ranges. A lot of times these are like 10 to the minus 6, 10 to the minus 9, 10 to the minus 12 dosage ranges. Again, the hormone ranges. It was found that many heavy metals like mercury and lead and cadmium at these very tiny doses, interestingly, actually stimulate your physiology. They actually have a healing effect, versus the larger doses do the opposite. And this is, of course, the basis of homeopathy. So a lot of those critics of homeopathy actually are maybe not taking hormesis into consideration. We have to be clear with homeopathy, though, that up to Avogadro's number, which would be 24x, or 12C, people have actually observed hormesis phenomena. What happens after that, we don't know. Essentially, Avogadro's number would be here, 0. And if you go higher, like a 30x, 30C, etc., you're going to be going to the left of the curve, and we don't know what happens out here. So I'm not going to talk about remedies above Avogadro's number. I'm going to talk about what we might call low-dose homeopathics up to Avogadro's number. Again, a lot of your hormones are actually in that range. It's been discovered that hormetins really encompass pretty much everything. So, good examples, exercise. A little bit of exercise to a degree, perfect. Too much exercise, we start seeing problems. Sleep, stress, alcohol consumption, studying. These all have hormesis-like effects, and these are known as hormetins. I mentioned heavy metals, even ionizing radiation. A little bit of radon, interestingly, stimulates your cells. Too much suppresses them. What we think is happening is at the very small doses, we create reactive oxygen species in the body, for example, from radiation, and that upregulates cell protective mechanisms. If you suppress those antioxidants with really large doses, or oxidants with really large doses of antioxidants, like high-dose vitamin C or other nutrients, you actually stop that signal. And very curiously, we're finding in the research data that if you eat foods with antioxidant-rich compounds, flavonoids and whatnot, which are absorbed at very, very low levels, that actually has a beneficial effect for prevention of cancer and heart disease and diabetes, etc. But if you take those same nutrients, concentrate them, eat them as a supplement, Lo and behold, you get the opposite effects. And we have at least animal studies, and now we have human studies too, looking at meta-analyses of antioxidant trials, that people are actually dying faster taking these concentrated antioxidants. Huge, huge problem here, because a lot of the naturopathic profession is really founded on using a lot of high dose nutrients, thinking that it's natural, concentrate more of it, you'll get a greater response. But that is not what the research literature is showing. And that is a really concerning trend that we need to really consider. So I'm going to be making a big point about that a lot of these sort of antioxidant type things and whatnot actually are maybe doing the opposite of what we really want them to do. A lot of drugs we know are actually working through hormetic mechanisms. So a good example is SSRIs. SSRIs, in the doses that we take them, actually act as a mild neurotoxin, which interestingly upregulates protective mechanisms in the hippocampus and the amygdala to protect those areas of the brain from, guess what? Cortisol. So curiously, SSRIs we think are working partly by protecting you from too much endogenous cortisol. And they do it through a hormesis mechanism. We think metformin is working in a similar way as well. One new theory about how herbs are working is known as Xenohormesas. And that is basically that herbs, the secondary metabolites in herbs, actually act as mild stressors to your system. And by stressing you, you increase those cell protective mechanisms. That has a healing effect, and that stimulates your physiology to heal in some way. You take that same substance, concentrate it, put it in like a drug, and now we get the opposite effect. You get an inhibition of your physiology. Now, importantly, I want to say in medicine, we might want that. Sometimes you need to inhibit, and that can be an important mechanism. So that's going to be part of the therapeutic order I'm going to talk about here last, is when we do which, and to understand when each is important. But generally when you eat, drink herbal tea or eat a diet, let's say you eat a diet high in flavonoids, the blueberries. Curiously, flavonoids are very poorly absorbed, less than 5%, and they're metabolized within a few minutes. So pharmacologically, they're kind of useless. And that's why a lot of pharmacologists are like, don't worry about those, because they're looking at the long-term drug effects and so forth. But interestingly, as your liver metabolizes the flavonoids, it dramatically increases production of endogenous glutathione, more than you'd ever get from an IV. And it's that glutathione that heals you. That gets into the blood, it goes out to the tissues and has an effect. So curiously, it's not the flavonoids which heal you. They're not antioxidant. In fact, we never absorb enough of those flavonoids to have an actual antioxidant effect. That's a petri dish phenomena. But in the human body, we metabolize them, but in the process of doing so, we increase our own endogenous antioxidants and other mechanisms. And so that has the healing effect. One final one I'll mention here is turmeric. Small doses, you eat it in the diet and so forth. Turmeric actually increases your bile flow, so you get rid of more junk from your liver. You increase hormone detox. It also, by putting more bile salts into your blood, it actually stimulates peripheral capillary circulation. So you get more circulation into your capillary beds. And if you have an area of chronic inflammation, we can kind of think of that as a cold inflammation, where there's actually usually very poor circulation into chronically inflamed areas. The turmeric drives up circulation, and that actually could temporarily increase acute inflammation, and that has a healing effect. Now, that's the low dose. If I take a high dose of turmeric, I concentrate it into capsules, I put them in phytosomes or doing intravenous. Now we're going to get the direct anti-inflammatory effect from turmeric, which again you may want, but don't just think that anti-inflammatory activity is what you want in everybody. A lot of times you want them to heal from that inflammation. Suppressing it is really only one approach, and that might not be the best long-term approach. So that's another example of a hormesis effect. We know hormesis applies to all the vitamins and minerals, and then for our purposes here, hormones are under hormetic control. Very important. Good example of this is testosterone. So endogenous testosterone in males we think is cardioprotective. It actually helps endothelial function. We know it improves the perfusion of blood into the myocardium, increases oxygen uptake, and so forth. So it has a protective role on the cardiovascular system. You now take exogenous testosterone, usually in supraphysiologic levels, So you're going way to the right of the curve here. We get exactly the opposite. We start to see that it stresses the cardiovascular system. You get worse cardiovascular outcomes, higher cholesterol, atherosclerosis, etc. So the same compound, different doses, two opposite effects. And this has created a real havoc in the research literature. And that's why people are like, oh, testosterone's good for you. Well, no, it's not. Yes, it is. And they're talking about different dosages, different application regimens. So what we really want to emphasize, and what I might argue is naturopathic endocrinology, is more getting your body's glands to secrete the hormones at the right levels, not artificially replacing them. When you do that, you usually give it in much higher doses than the body's used to. Puts extra stress on the body that way. And you also don't get those nice circadian rhythms and so forth when you provide it exogenously. So interestingly, although I teach endocrinology, deal with a lot of it in the clinic, I'm not a huge fan of hormone supplementation, with some exceptions. Thyroid's a big one that we often do a lot of. Insulin, of course, if you're type 1 diabetic. But a lot of sex hormone replacement, all that kind of thing, we have to be very careful about when that's really needed. And we'll talk about those cases as we go. All right, so that is hormesis, and here is the mechanism. So we can really talk about the two different effects. We have low-dose effects that support your physiology, and they support actually healing of the tissues. And again, that term, I'll talk more about it later, is called salutogenesis. That's supporting the conditions for health. And then high-dose effects would be addressing pathogenesis, like suppressing inflammation, antipyretic, anti-inflammatory, etc., etc. So the anti-approach is the high-dose approach. So we're going to look at both of those, and but I just think it's important at this point to understand that basic concept. All right, so let's look at the three basic types of hormones. We actually have really two. We can say they're hormones derived from amino acids and those derived from steroids. Now, in terms of amino acids, we have the amide hormones, and they're all derived from tyrosine, and that includes your catecholamines, epi, norepi. They're secreted, of course, norepi from your sympathetic nervous system, but both are secreted by the adrenal medulla directly into the blood. They're water-soluble, don't need a hormone transport, and they're carried to the tissues. We can't take them orally because your digestive enzymes, your proteases, would break them down. Thyroid hormones are also derived from tyrosine. The thyroid gland secretes primarily T4, levothyroxine, and your tissues then convert the T4 into T3, triiodothyronine, which is the active form of thyroid. We're going to look at that in detail, so this is just to kind of lay this out at this point. Now, these are not very water-soluble, so they have to be carried in the blood, bound to a transport protein, primarily thyroid-binding globulin, a little bit of albumin as well. So that is how the thyroid hormones get around this way. Now, the catecholamines act on their own receptors, of course, alpha and beta receptors. You should know that by now. We, of course, have two major alpha receptors, alpha-1, alpha-2s, and the betas, beta-1 on the heart, for example, also in the kidneys, beta-2s on the lungs, beta-3 on adipose. And binding of the catecholamines to these receptors stimulates intracellular adenylyl cyclase, which increases cyclic AMP, and that acts as a second messenger to stimulate all sorts of intracellular activity. So that's the amide hormones. They are very fast acting. Versus thyroid receptor is, interestingly, intracellular thyroid hormones are transported through the membrane, T4 primarily. In the cell, there are enzymes called deiodinases, which convert T4 to T3. And then the T3 binds to the thyroid hormone receptor, which often hangs out in the cytoplasm. Once it binds, it translocates to the nucleus, and then it activates gene transcription. So that's how thyroid hormone works there. The other major class of hormones would be the peptide hormones. These are longer strains of amino acids. These are all water-soluble, and they comprise most of the hormones, like from the hypothalamus, your insulin, parathyroid hormone, calcitonin. All of these can range from being very small, just 3 amino acids in length, for example, to very large proteins, and they're often glycoproteins. They have sugar moieties attached to them as well. They don't need a transport protein because they're water soluble, and they, of course, can't be taken orally because you would digest them. So we'll see all these would have to be injected if you're going to use them as therapy. They bind with protein receptors on cell membranes. These are typically G protein receptors. I'll review that here in just a second. And the binding of the hormone to the G protein initiates adenylyl cyclase again, and this stimulates intracellular signaling. And so that is the G protein. And then finally we have steroid receptor, steroid hormones, which are all derived from cholesterol. And these would be what a lot of people think of as hormones. So things like your cortisol, DHEA and testosterone, pregnenolone, all from your adrenal cortex. And then your sex hormones, the estrogens, and there are 3 different types. The main one is estradiol, E2, but there's also estrone, E1, and estriol, E3. We'll look at those later. Progesterone, testosterone, and then vitamin D I'm putting here is also derived from cholesterol. Again, it's a kidney hormone. These are not stored, unlike the peptide and the amide hormones, which can be stored in granules in the cells before being released. These actually have to be synthesized on demand and are released immediately. So we don't store up any of these within the cells. They are lipid soluble, so they all need transport proteins to be carried in the blood. So a big one for estrogen, for example, and testosterone is sex hormone binding globulin. Now, all of these transport proteins, I should say as an aside, are made in the liver. So the liver has a major role of regulating hormones by binding up these hormones. In fact, most of the hormone in the blood is bound. Bound hormones, though, are not active. So we have to dissociate the hormone from the binding carrier, and then that diffuses into the tissue for it to be active. So only a very small amount of hormones are actually in the free form. And we'll talk about that when we come to hormone measuring, when we look at cortisol or these different things, like especially estrogen or so forth. We're going to have to look at the difference between free versus the bound forms of these hormones. Again, free is only what's going to be available to the tissues. In some measuring techniques, like salivary hormone testing, many proponents argue that they're directly measuring the free hormones, and that's going to be more important than the plasma testing, which often is going to measure mostly the bound hormone, and we need to differentiate those. So we'll come back to that as we go. The steroid hormones then usually are able to get right through the cell membrane. They diffuse right across. They don't need to be transported. And then they bind to steroid hormone receptors, which are found either in the cytoplasm or the nucleus, and then that initiates gene transcription. Now, interestingly, estrogen, progesterone, and testosterone have also been shown to have cell surface receptors, and those have been only recently discovered. And we are finding that steroids actually have an ability to actually work on cells genomically. In other words, they can trigger gene transcription and translation. Or non-genomically, they directly influence the intracellular signaling, and that is able to influence cell metabolism. So that's an additional effect. The non-genomic effects are usually much more rapid. The steroid hormones, generally when they work in the genomic way, by initiating gene transcription and translation, takes several days to actually have an effect. So interestingly, we now know that the sex hormones can also have a more immediate action via the cell surface receptors. All right, so those are the 3 major types of hormones and their major receptors. I'm going to just go into the receptors in a little bit more detail here. Okay, so this should be basic review from physiology, but in case you've forgotten, there are 4 basic types of receptor classes. The first type is going to be where an agonist binds to a cell receptor, and that opens up an ion gate. So these are ion-gated ligand channels. So basically, a good example would be like acetylcholine on the neuromuscular junction binds, causes sodium to influx into the cell. These act very rapidly. These are more common in the nervous system versus the endocrine system. The second type would be the G protein, which I talked about. That's a transmembrane protein which weaves in and out of the cell membrane 7 times. The ligand comes out here, and that could be a hormone, a peptide hormone, binds to the G protein. That causes a conformation change in the protein that activates the G protein inside the cell, and that generates second messengers, and that's what activates the cell signaling. So that's G protein. The third is a tyrosine kinase receptor. The insulin receptor is one of these types, where the ligand, the agonist, binds to the receptor, and that causes phosphorylation of tyrosines on different signaling molecules within the cell, and that activates cell signaling. And then the final type would be the steroid hormone receptor, where the agonist is the steroid hormone. It goes right into the cell membrane, binds to the receptor, which again is often the cytoplasm or the nucleus. It's then transported into the nucleus, and that activates gene transcription and translation. So those are the 4 different types of cells. The fastest are on the left here, the slowest are on the right. Here's just a little diagram of how a G protein works, G protein receptor in more detail. Here's the ligand, maybe a peptide hormone binding here to the G protein, activates the G protein, which then comes over here, which activates adenylyl cyclase. Adenylyl cyclase then catalyzes the conversion of ATP to cyclic AMP, and then that is going to activate different protein kinases. Remember, a kinase is an enzyme that phosphorylates proteins, and that pretty much turns that protein on, and that activated protein kinase now will have different intercellular activities. So that's the classic pathway of a G protein receptor. And here is the steroid hormone receptor. So the steroid hormone comes in, crosses easily the plasma membrane by diffusing through, binds to the receptor protein, usually in the cytoplasm, is translocated into the nucleus, where it then acts on DNA to initiate transcription into messenger RNA of a certain protein, and then that messenger RNA in ribosomes is used to make new proteins, and that protein then has some effect on cell metabolism or is excreted from the cell, and so forth. Again, this is the slowest type of receptor, and it often takes 2, sometimes even 3 days for this to really upregulate. And finally, here is the insulin receptor, an example of a tyrosine kinase receptor. Insulin receptor is complicated. There's an alpha portion on the outside, beta portions on the inside. These are protein chains. The insulin binding to the alpha portion stimulates different signaling pathways through phosphorylation of tyrosine kinases. There's, for example, something called the MAP kinase pathway and the PI3K pathway. And then the PI3K pathway actually causes more of these little transporters called GLUT4s. If this is a muscle or an adipose cell, it inserts these in the surface of the cell, and that actually allows glucose to come into the cell. So this is how insulin gates glucose outside the cell. This is how it allows glucose to get into the cell. Again, this is only for specific types of cells like muscle and adipose. Many cells can actually get glucose into them without insulin, so they're insulin independent, and they have different mechanisms through which this goes through, and we'll talk about that later. But this is one example here of insulin. Notice that insulin also has all sorts of other intracellular activities, and these regulate metabolism. So you can see that a single receptor can have multiple intracellular effects. One final thing here I'll say, we've all heard of this term insulin resistance, and that is a very complex area because that can involve problems either here at the receptor, but also through any one of these pathways. And so there are actually literally, and this is a simplified diagram, there are hundreds of intracellular steps here, and there could be a breakdown at any of those levels. So as we'll see, insulin resistance is a very complicated thing. There's no one specific mechanism necessarily to target. Now, if we look at the endocrine glands, interestingly, they're pretty much, again, 7 basic glands, and they're all arranged along the vertical axis of the bodies. In fact, if you've seen these old diagrams of the chakras from the Ayurvedic tradition, they really correspond to where all the major endocrine centers are. And what's very interesting is around each of the glands, there's a major nerve center. So if we talk about the neuroendocrine system, really there are 7 basic neuroendocrine organs, and that's what I'm calling the neuroendocrine centers. One very curious thing also is around many of these centers, there are lymphatic bundles. And the lymphatic bundles have T cells and B cells and so forth that are all being programmed by the hormones as well as the nervous system. And I'll get more into that, how the immune system is actually programmed by the neuroendocrine system. So for instance, if you're very stressed all the time, you eat a bunch of different foods, in the spleen, you'll actually become more sensitized to reacting to foods if your sympathetic nervous system is high. If you have more acetylcholine from a relaxed state, you're more in a parasympathetic state, the same food will now trigger a Th3 or tolerance response in the spleen, and you won't react to that food. So this will play a big role clinically, where a lot of our food sensitivities actually are not from the food per se, but rather from a misprogramming of the immune system, which can be modulated by changing the neuroendocrine system. And so that'll be really important when we look at different therapies. So if we go through these neuroendocrine centers, we can start with the pineal gland and its major secretion, melatonin. There's many other secretions too. I'm not going to talk about that here. And we have the cerebral cortex around that. Again, the pineal's right at the back of the midbrain. In the next major set of videos, you'll go through the pineal gland and look at that in more detail. The hypothalamus and the pituitary, both anterior and posterior, these secrete a whole slew of different peptides. So growth hormone, thyroid stimulating hormone, ACTH, prolactin, FSH and LH, the lipotropins and endorphins, as well as oxytocin, vasopressin. These also are part of the brain, just below the optic chiasm. And then the thyroid down here in the neck. We have the thyroid gland, which secretes primarily T4, some calcitonin, and the parathyroid, which secretes parathyroid hormone. This is surrounded by the sympathetic, the cervical chain ganglia. And then we have the heart, and we can put the thymus in this region as well. Here we have a cardiac plexus, both innervated by sympathetic and parasympathetic nerves, branches of the vagus. And then we have a third neuroendocrine center here in the epigastrum, the solar plexus. This is the pancreas, with even the liver and the small intestine. All of these are paired together in a way. And there is the celiac ganglia here with the vagus branches as well. And then we have a second center down below that with the adrenals and the kidneys and their innervation, and then the gonads themselves. So these 7 centers, and actually the question mark, this should actually be pelvic plexus. So all these neuroendocrine centers, we can say these are the 7 major areas that regulate your internal physiology. So clinically, I'm often looking at which one of these centers is actually weakest, which is most disturbed. And we'll talk later about how we can do that. We can measure that through blood tests. We can kind of get a sense of that through a patient's history and through some other little exams we can do. But that would be kind of a way of organizing all this information so it doesn't get too confusing for you. All right, so once hormones are secreted from the gland, how are they regulated in the plasma? We have to maintain fairly steady states of these hormones in the plasma. Again, some of them have circadian rhythms, many of them actually do. So a good example is TSH, thyroid stimulating hormone. It's actually highest in the morning hours, and then it goes down in the afternoon. So it's going to vary during the day. But there are several factors that actually regulate how much hormone is in the blood at any given time. And of course, that's going to determine how much is able to get into the tissues to have an effect on the target organs. There are a lot of what we might call humoral or glandular factors which regulate hormone biosynthesis. That would include the presence of other hormones, so specifically stimulating or releasing hormones, for example, from the pituitary. So TSH stimulates the thyroid to release the thyroid hormones, ACTH, the adrenal hormones, and so forth. These are called tropic hormones. Notice that word tropic. There's 2 words here which often get confused. There's tropic and there's trophic. Tropic means it's like a peptide from the hypothalamus. It stimulates another gland to secrete its hormone. That's a tropic hormone. A trophic hormone, on the other hand, is any hormone that stimulates growth or proliferation in tissues. So a lot of people get those terms confused. Trophic, again, stimulating growth or differentiation, versus tropic, more like a signaling molecule to tell a gland to secrete another hormone. Different concentrations of ions and nutrients and binding globulins also help regulate the hormone concentrations in the blood. Cytokines feed back on the endocrine system, so we'll have to look at that and how they can influence hormone secretion. Different nutrients like availability of cholesterol. In fact, it's LDL cholesterol, which that lipoprotein which carries cholesterol from the liver to the adrenal gland and the gonads to make sex hormones. So that's how those glands get their cholesterol. Turns out they can actually make a little cholesterol themselves, but it's mostly from the LDL. Presence of iodine for thyroid, different cofactors. So a lot of these hormones, the main activity is the enzyme activity within the gland. So a good example is the adrenals. Pregnenolone is the starting hormone for all of your steroid hormones. It comes directly from cholesterol. And then through different enzymes is converted into all the different hormones. A lot of people think, oh, if you're low on different adrenal hormones, let's just give you pregnenolone, get your pregnenolone levels up, and you'll make more of these other hormones. Turns out it's not that simple, because you can have lots of pregnenolone, but if those other downstream enzymes are not active, and many of those enzymes require different cofactors like copper or zinc or magnesium to function, or different coenzymes like different B vitamins. If you're deficient in those, then those enzyme activities simply won't work, and you won't convert that pregnenolone downstream. So that's more important than the actual levels of the hormones, is the level of these enzyme activities. The nervous system also is a major integrative player in the endocrine system, and we'll have to look a lot at the hypothalamus and how it does that. And that's very much influenced by your mental emotional activity, environmental changes like light and temperature, your endocrine rhythms, so that would be those different chronobiological rhythms I spoke about, and then the presence of negative or positive feedback cycles. So that's one major step in the regulation of plasma hormones. Another step in regulating plasma hormones is what we call precursor processing. And a good example of this is, for example, when the beta cells in the pancreas make insulin, they actually make a what they call proinsulin, a precursor to insulin, which is a large molecule. And that's actually, oops, this diagram is backwards down here. But proinsulin actually is a large molecule with 3 peptide chains. There's a A chain, there's a B chain, and there's a C chain. And that actually has to be modified where we actually cleave out the C chain and put the A and the B chains together, and that's insulin. So that's called precursor processing, and that happens usually in the endoplasmic reticulum or Golgi apparatus of the different secreting cells. Another example is ACTH. That's a really interesting one. It actually starts in the secreted by the anterior pituitary gland, and the precursor molecule is called POMC. And POMC fragments into all these different hormones like ACTH, but also beta-lipotropin, and those further fragment into things like endorphin, beta-endorphin, melanocyte-stimulating hormones. So when you release ACTH, you also release all these other things like endorphins and so forth. And so that's curious. As part of the stress response, you actually release endorphins to help you deal with pain and those kinds of things in that process. So that's one important step, is precursor processing, and anything that inhibits that is going to disturb hormone levels in the plasma. Hormone released, many hormones are also stored. Again, the non-steroid hormones can be stored in granules, and they're released in response usually to second messenger system, so ligands have to bind to that cell to stimulate the release, or the nervous system has to activate. So that's another little mechanism there. They can regulate plasma hormone levels. Another mechanism that regulates plasma hormone levels would be the binding globulins. So again, many lipophilic or hydrophobic hormones have to be carried by carrier proteins. Albumin is kind of a carrier for everything, so that can sometimes carry proteins, but thyroid has its own specific thyroid binding globulin. Cortisol has its own corticosteroid binding globulin. Sex steroid binding globulin binds estrogen and testosterone, and that's actually sex hormone binding globulin. And that the hormone bound to that, so if we have all this bound up hormone, it actually serves as a reservoir, sort of a pool, and then a little bit of that diffuses into the free hormone, and that gets into the tissue. So usually like 98, 99% of a hormone is usually bound, and only 1 or 2% is free in the tissues. And there are different lab tests. Some of them test for the total, which would be both the bound and the free. And some tests just look at the free form. So we'll have to differentiate those. Good example is thyroid hormone testing. Total T4 would look at bound and free. And the total levels are going to vary by, for example, different levels of thyroid binding globulin. And that's going to really depend on different liver metabolism. So, for example, If we have problems of fatty liver again and so forth, that can actually create problems with thyroid binding globulin. High estrogen levels will actually raise thyroid binding globulin. That way most of your thyroid will be bound up and only a little bit of it will be free. So that actually makes less thyroid available to your tissues to be active. And so that could be a big issue too, is other hormones regulating the binding globulins. The more of that binding globulin, the less of the free hormone you're going to get in the tissues. So it's going to slow the metabolism of that hormone, keep more of a plasma pool of it, but also if that carrier protein is too elevated, it might actually make less available to the tissues. An interesting phenomenon that happens, for example, as men age is their sex hormone binding globulin goes up and they make less testosterone. So interestingly, the testosterone they do make is mostly bound up and they get less and less free testosterone to the tissues. So that's another example of where that can play a role. Finally, another big process that regulates hormones, which are going to be very important as we look at different clinical conditions, is hormone metabolism. The liver is the primary organ that metabolizes most hormones. So there's different effects of metabolism. Some of the metabolic activities actually, like the conversion of 25-hydroxy vitamin D to 125, make it more active, or androstenedione into testosterone, that makes it more active. But usually we think of hormone metabolism as inactivating the hormone, degrading it into inactive metabolites, which are excreted through the bile or the urine. And that's going to depend on liver detox. And I'm going to go over that in the next slide because it's important to review that for this discussion. And then many of those metabolites, again, will be eliminated through the bile or the urine. Thyroid hormones are deiodinated. You take the iodine off of them and that inactivates them. And there are enzymes that do that. All right, so hormone metabolism is going to also be sort of the end stage of what regulates the different hormone levels in the blood. So as I said, I want to say a little bit about liver detox here, because I think a lot of people either forget this or they get a little bit confused. The liver actually, a lot of people talk about how the liver breaks down substances to detox them, and that's actually not accurate. The liver, interestingly, actually makes larger compounds out of the starting molecules. It's primarily an anabolic organ. It's building. And the reason it does that is to make a molecule more water-soluble than it was before, and that makes it less toxic and more easily excreted. So let's look at the different liver detox processes here. So the first one is going to be where we start with a substance like a drug or a phytochemical or toxin, or in our case hormones, and it's going to go through two stages of detox. Phase one occurs not just in the liver. That happens in the liver, the gut, a little bit the kidneys. But this happens through what are called CYP450 enzymes. There's about 100 different CYP450s in the body, and about 6 to 10 of them are really important pharmacologically because they metabolize different drugs and so forth. And these are concentrated, usually they are found in the endoplasmic reticulum or different parts of the cytoplasm. And they essentially are going to convert lipophilic molecules into more polar molecules, essentially by introducing a hydroxyl or an amine group. So that's going to create an intermediate. Unfortunately, the intermediates are often more toxic than the starting substances. So I'll show you, for example, what happens to Tylenol, paracetamol or acetaminophen, and how the intermediate is actually more toxic than the original starting compound. Then it's going to go through phase two, which is only in the liver. And that's a conjugation reaction where a chemical group will be added to that intermediate. And it usually adds it at the place where that hydroxyl or the amide group was added. And this is going to make the substance essentially non-toxic, and the body can then excrete it through the bile or the urine. There are 6 major phase 2 pathways. So glucuronidation is sticking on glucuronic acid, which is a sugar. So that's, for example, when you hear about bilirubin. Unconjugated bilirubin is the bilirubin that comes out of the spleen after your hemoglobin, your heme breaks down. It makes it into hemoglobin, into bilirubin. That's carried by albumin, primarily to the liver. And then the liver conjugates it with glucuronic acid, so we get conjugated bilirubin. But also many hormones are detoxed that way. Sulfation is an SO4 group. That's another detox pathway. Again, many hormones are detoxed that way. Methylation, we have a lot of talk about methylation. It's addition of a CH3 group. And that also is how, for example, a lot of estrogen metabolites are metabolized in the liver. Acetylation, adding an acetyl group. Amino acid conjugation, that's usually with glycine, the simplest amino acid. And then glutathione conjugation. So these are the 6 phase 2 detox pathways. We don't talk about this much anymore, but I was a student back in the 90s in the naturopathic profession. We talked about pathological detoxifiers. And these are people who, for various reasons, either nutritionally or genetically, are not able to manifest these detox pathways adequately. And so they're not able to clear the intermediates as quickly. And so they can allow these different things to build up in the body, often to toxic levels. And so we could actually do tests, for example, have people swallow caffeine. And in the process of swallowing caffeine, you would... you could actually measure how long it would take for it to leave your body, and for some people that was a couple of hours, and others it was actually 2 days later they were still detoxing the caffeine. So we don't do those tests anymore, but they kind of showed us that there's this tremendous variability to how people are processing through the liver through phase 1 and phase 2. Here is another little diagram of that down here. And what's important here is that, we can actually support these pathways through using different nutrients. And so it's possible by using different nutrients to actually speed up different pathways. So we might want to speed up, for example, in the case of estrogen detox, we might want to speed up using different sulfur compounds to improve sulfation, or using methyl donors to improve methylation, and so forth. And we'll get into that when we look at estrogen metabolism much later. So that's a little bit there on liver detox. Here's just a couple of other examples of detox. So here is again acetaminophen, Tylenol, where when it goes through the cytochrome P450, specifically cytochrome the 2E1, that it becomes a compound known as NAPK. N-acetyl P-benzoquinone imine. This is very toxic, and this is actually where some people can develop liver failure within a couple of hours of ingesting Tylenol if they're not able to take NAPK, conjugate it with glutathione, and remove it through the bile of the urine. Once it's conjugated with bilirubin, it's not toxic anymore. So this is where we can actually load a person with NAC to N-acetylcysteine to try to increase their glutathione. That's often done now in the hospitals using IV NAC if a person comes in with liver toxicity from NAPK, and that hopefully can help a person detox that way. So that's one example of where phase 1 and phase 2 come into effect. I'll say a lot of the additives to cigarettes, we know the starting material often is not particularly toxic, but once it gets metabolized through phase 1, it becomes quite toxic. And if a person has very slow phase 2, they're going to have more damage from those intermediates than maybe a person who's able to clear that more quickly from their system. Here's another example using estrogen. So estrone is one of your types of estrogen. It can be interconverted with estradiol, the main type of estrogen. But there are 3 basic types of metabolites of estrone depending on which CYP450 enzyme system it goes through. So if it goes through CYP1A1, we get what's called 2-hydroxyestrone, which can be further methylated through an enzyme called COMT into 2-methoxyestrone, and that actually is non-toxic, and we think it actually has a slight protective role if it gets into the blood on binding to estrogen receptors and blocking excess estrogen from binding to them. Unfortunately, if the same estrone goes through CYP3A4, it becomes 16-alpha hydroxyestrone. And that one itself can usually, we think that might be carcinogenic. Most people, though, can convert it directly into estriol, which is non-toxic. So that one's not too much of a problem. But it's the CYP1B1, which can become 4-hydroxyestrone. And that goes into what are called 3 to 4 quinones. These estrogen quinones we think are quite carcinogenic. And so people have a much higher potential incidence of breast cancer, uterine cancer, and so forth from those estrogen metabolites. So this is where we can look at different substances like DIM, diindolylmethane from I3C. This is the indole 3-carbinol. This is a sulfur compound from brassicas. It can actually help to push the pathway more towards the 2-hydroxy estradiol and estrone. So it pushes this first pathway here. And so we get the non-toxic metabolites. And we'll talk about many others as well. We can increase methylation donors to try to help to methylate these compounds more quickly, and they become non-toxic. So that's another example of how liver plays a tremendous role in regulating hormone metabolism. Now, I mentioned several times that one of our major integrating centers in the brain is the hypothalamus. This is part of the diencephalon. We'll go into this in detail when we look at the pituitary gland. This is part of the middle of the brain, part of your emotional brain structures. And this is really where we have the hypothalamus connected by a little stalk to the pituitary. And the hypothalamus secretes different hormones which get into that stalk. They tell the pituitary cells to release their own hormones, and then that signals to downstream glands to release their hormone. So a good example of that is the Hpa axis. So the paraventricular nucleus in the hypothalamus releases a hormone called CRH, and that is going to tell the anterior pituitary to release ACTH, and that gets into the bloodstream and tells the adrenal cortex to release cortisol and other glucocorticoids. And then as their levels build up in the blood, they feed back to the hypothalamus, and that turns down the cycle. And so that's known as the Hpa axis. And a lot of things like stress, inflammation, insulin resistance, and so forth can all trigger this pathway to fire. And we'll look at all those details as we go. So that's one axis, Hpa axis. But we also have one for the gonads. So we have a hypothalamic pituitary ovary and testes axis, and that's through LH and FSH, and then their hormones, testosterone and estradiol. And then the HPT axis, hypothalamus pituitary thyroid axis. And then there's also one with growth hormone. Growth hormone, we'll look at that in detail. It's interesting because it actually in and of itself has very little effect on the body. There's some, but it's mostly when the growth hormone gets into the blood, it tells the liver cells, hepatocytes, to secrete IGF-1, insulin growth factor 1, and that's what really causes growth in the muscles, in your ligaments and tendons, your bones, and so forth. And that's known as the somatomedin axis. So these are the four primary regulatory axes from the hypothalamus, and very important to understand as we look at different disorders of the pituitary and the other glands. And finally, I just want to say a few words about the hypothalamus before we move on. We'll look at this in a little bit more detail in the pituitary section. But really, as you probably remember from your basic organ systems, the hypothalamus, of course, is a major regulator of body temperature, thirst and appetite, water balance and blood pressure, your circadian rhythms, and GI stimulation. This is really where the sympathetic, parasympathetic nervous system is highly regulated and balanced, and so forth. And then through the secretions in the pituitary, we have a connection with the endocrine system. So this is the primary neuroendocrine organ here, up between the eyes, in the forehead, that regulates all these different activities. Couple of important hypothalamic areas. This is usually board exam questions to maybe know about. The lateral areas of the hypothalamus, this is really your hunger centers. Destruction of these areas results in anorexia in laboratory animals or people that have strokes in these areas. And this area is inhibited by leptin. Leptin is secreted by adipose tissue. When the adipose becomes too filled with fat, it signals leptin, and it should tell the hypothalamus to downregulate appetite. But so the lateral area, if it does its job, it should trigger hunger. Again, leptin would inhibit that. The ventral medial areas of the hypothalamus are our satiety centers, and destruction here results in hyperphagia, and this is stimulated by leptin. And then the posterior hypothalamus is really where your sympathetic nervous system is more regulated, and your sympathetics, as they engage, really increase catabolism and they warm up the body. So we might think of this as our heating center. And then our anterior hypothalamus is your more cooling center related to the parasympathetic nervous system. And there's a very important gland right here called the suprachiasmatic nucleus, in this region here. It's called suprachiasmatic. It's hard to see in this picture, but this is the optic nerve that comes right through here, and this is where the optic nerve crosses right between the hypothalamus and the pituitary. And there's some nerve bundles that go up from the optic nerve into this region. And those nerves are carrying information from cells in your retina called ganglion cells. These are different than the rods and the cones. The ganglion cells, interestingly, are sensitive to blue light. And so when you are seeing blue light, either on your computer screen or through the blue sky, it's activating, it's triggering these nerves, and that actually is turning on and regulating the suprachiasmatic nucleus. The suprachiasmatic nucleus really has an internal clock, and this is really what regulates our 24-hour rhythm and sets the circadian rhythms for all these other hormones. Interestingly, if you keep a person in the dark or in a cave, not exposed to the blue light, they will naturally synchronize to about a 25-hour rhythm, not a 24-hour rhythm. Curiously, that's a lunar cycle, a lunar rhythm, versus the 24-hour is a solar cycle. So the suprachiasmatic nucleus keeps us timed to the light of the day and sets our internal hormone rhythm. So that'll be a very important center we'll need to remember. All right, so that wraps it up for a general introduction to the endocrine system. In the final video, I'm just going to go very briefly into just some discussion about treatment of endocrine disorders from the naturopathic perspective, and then we'll jump right into the different glands, starting with the pineal gland.